a colleague of mine, Manuel Rausch, from the University of Texas at Austin. Manuel Rausch got his PhD at Stanford University, and we spent some time together there. And then after spending some time in industry, he did a postdoc in Yale, and before joining the University of Texas at Austin. And he's a very productive researcher and also has many awards, like the American Heart Association Career Development Award. And yeah, so we get the pleasure to learn a little bit more about the image and experiment-based modeling of the forgotten right side, the right ventricle. So I'm gonna stop sharing my screen, and actually I'll ask the staff to please record the seminar. So we'll just maybe pause for a second or two. All right.
Okay, can everyone see my screen?
Yes.
Awesome. Adrian, I truly appreciate the introduction and lying about my number of awards. You're a true friend. I would like to thank the entire staff for putting this together a second time for me, and apologize to everybody who's here a second time. Hopefully everything goes smoothly, and I get to talk about my work on image and experiment-based modeling of the forgotten side. Before I start, I'd like to really appreciate the hard work that my students did for this, as well as my collaborators. Some of them are here today. And yeah, thank you all for being here during these crazy and busy times. So my lab really focuses on soft tissue disease. And during my PhD training, my industry time, and my postdoc, I sort of trained in experimental and numerical methods. And I tried to bring this all together toward this goal to understand and hopefully one day contribute to the treatment of various soft tissue diseases. My specific focus has been recently on the forgotten side. And so that's in the title of my slide, and you may be wondering what this means. And specifically, when I talk about the forgotten side, I talk about the right side of our cardiovascular system. So generally speaking, when you hear about the cardiovascular system, you hear about the heart valves, and our vessels. And so generally speaking, they're divided into two sides. The left ventricle, the mitral valve, the arterial vasculature are sort of representatives for the left systemic side that supplies oxygen to our cells, and then the blood returns to the right side, to the right ventricle, the tricuspid valve, and the venous vasculature. And there are physiological as well as historic reasons that the right side was just not studied in as much detail, and has therefore been dubbed by us and others as the forgotten side. Recent developments within this COVID-19 epidemic have really sort of highlighted the importance once again of the right side. For reasons that are not entirely understood at this point, the right side seems to be disproportionately affected by COVID-19, including the right ventricle, as well as venous thromboembolism. And I think it's just a good reminder for us to make the forgotten side unforgotten. So subject of today's talk are these three projects that I work on: right heart mechanics or right ventricular mechanics, tricuspid valve mechanics, and thrombus mechanics. So we combine imaging and experimentation to do modeling work. And I start with talking about the right ventricle. But before I do so, or before I talk about my work about the right ventricle, I want to do a quick sort of anatomic review, given that not probably everybody looks at the right ventricle and the right heart on a daily basis. So this is a 4-chamber view of the heart. And we see the left side over here, and then the right side over here. And you may be confused given that the right side is really technically on the left side. It's described as per the patient's view or your personal view. So if you look down, the right ventricle would be on your right side. And basically the blood returns from your body into the right atrium, which is the chamber up here, then flows through the tricuspid valve, which is the subject of my second part of this talk, into the right ventricle, which we see here. The right ventricle contracts and pumps the blood into the lung, from where it returns to the left ventricle. The two ventricles, the right and the left ventricle, are separated by the interventricular septum. And so I specifically look at right ventricular myocardium, and myocardium really just means heart muscle. And it's an interesting and complicated material. And what we see here is sort of a cartoon version, where we see the muscle fibers of the really right ventricular muscle. And so you can see that this is a highly organized tissue. That means it also has a direction, a preferred direction, or an axis of symmetry. It's a heterogeneous tissue. We have the epicardium on the outside, the endocardium on the inside. In between the tissue is layered, and its material axes actually change, and we'll be talking about this. Together, these properties and these sort of structural phenomena, they lead to myocardium being an anisotropic tissue, being heterogeneous, reacting nonlinearly under deformation, showing signs of viscoelasticity. In vivo, the material can actively contract, and in disease, long-term, it can grow and remodel. So it's a pretty complicated material, and that makes it a challenge to study it. So why study the right ventricle? I mean, first of all, there's relatively little known about the right ventricle, which is, again, why we call it the forgotten ventricle. We don't know that much about function, structure, or disease. One of the diseases that I'm specifically interested in is a pretty interesting phenomenon in which patients that receive what's called an LVAD, a left ventricular assist device, so we see an image here, they tend to develop RV failure, so right ventricular failure, after we implant this device, whose function is to support the left ventricle in disease. So you can imagine this really as a pump that pumps blood from the left ventricle into the aorta. So it bypasses the left ventricle and is supposed to take work off of the ventricle that would otherwise be failing. So now these patients, for some reason, develop this right heart failure after they get this left ventricular assist device. Of course, you know, that's counterproductive to the entire idea of helping the patient. It is likely that wall stress could be a predictor for this kind of failure, given that wall stress is correlated with energy expenditure. So the idea is essentially that the ventricle, the muscle, produces tension, and to do so, it needs energy, which depends on oxygen. So in other words, if we need to produce more tension, we increase the stress, we need more oxygen. And if the ventricle fails, that is often related to the lack of oxygen being provided. So wall stress could be an interesting predictor that hasn't really been looked at, in particular when looking at LVAD-induced right ventricular heart failure. So our motivation here was to say, we really need a high-fidelity computer model to back out these wall stresses that aren't accessible through imaging or sort of standard clinical techniques. And to do so, we essentially developed this pipeline, which I'm showing here on the left, and I'm using to kind of talk you through our project. So you see there's an in vivo component, an in situ and vitro component, and then this sort of modeling part. We start out by studying sheep. So ovine hearts, you see here an image. So this is an actual sheep heart. Down here, you see the left ventricle. Up here, you see the right ventricle. And its geometry is kind of awkward. It kind of wraps around the left ventricle, and you see that geometry in the subsequent slides. And on this right ventricle, we implant these sonomicrometry crystals. These are essentially piezos that we can track throughout the cardiac cycle. So they serve as fiduciary points. And you'll see what we do with them down the line. So we can record now these marker coordinates in the beating hearts of these sheep. The chest can be closed if necessary, and we can record these data points at about 250 hertz, so at a pretty high rate. Subsequently, we remove these crystals, sacrifice the animal, and replace them with MRI markers. So what you see here are actual MRI markers in the positions where the sonomicrometry crystals were before, and then we can do an anatomic scan. So this is a geometric reconstruction of a sort of anatomic MRI scan, your standard scan. And then next, what we can also do, and I did this with the help of a colleague of mine, Jeff Lucy, who's on this call actually, we also did what's called a diffusion tensor MRI. So this is an MRI technique that allows you to track the anisotropy of your tissue, in a sense, non-invasively. Of course, at this point, the ventricle is explanted, so I wouldn't necessarily refer to it as non-invasive, but we don't have to dissect the tissue. We don't have to do histological slides, for example. And so what you see here are really reconstructions of the microstructural orientation of this muscle throughout the entire domain that we previously imaged with our anatomic scan. Once we're done with this, we transport the heart into my lab, and we cut out small tissue sections between 5 and 10 millimeters in edge length. And we glue them onto little pin stubs and then do simple shear experiments. So we basically have 2 pin stubs, and we shear them, and we measure with a triaxial force sensor the forces in all 3 directions. And so we can now shear that in one direction, in another direction, and then do constrained uniaxial tensile and compression tests. We remove that same sample, carefully remove the glue, reorient the sample, and repeat those 3 tests, and then we do this one more time. So we test these samples under 9 modes, and we measure forces in 3 directions when doing so. So at the end of this test, we actually have 27 force displacement curves on this one tissue cube. After that, we fix that tissue. We now conduct a sort of standard histological analysis, which means that we submit this tissue to a professional service that stains them, and that highlights the microstructure of this tissue. We can use very standard sort of imaging modalities. This is an NIH funded code called ImageJ that gives us the fiber orientations now of this muscle. So we get the microstructure of this muscle at a really high resolution, right? So we have a bar of 100 micron here. And we actually did this in 10 slices throughout the cube. So we get a spatial resolution of somewhere between 500 micron and 1,000 micron in thickness, and then a very high resolution in plane. And based on these data, we can identify the probability density functions of these fiber orientations. And with these 10 slices now, we can interpolate the entire microstructural orientation throughout the thickness of one of these cubes. So section 1 through 10 is essentially a cut from the epicardium to the endocardium. And you see that this is not a constant angle, but it's sort of slightly angled. And also importantly, this looks very different from the left ventricle, and just reminds us that we have to do a lot of work on the right ventricle because we can't simply extrapolate the behavior of the left ventricle to the right ventricle. There has been fantastic work being done on the RV, but much less so than on the LV. And one of our fundamental questions was, what is a good constitutive framework for the right ventricular myocardium? We had this opportunity really to collect this very rich microstructural data, but we didn't know how necessary this was. And in the future, ideally, we wouldn't want to spend thousands of dollars on histology for each of these cubes. So we actually did a case study in which we used a very simple approach to reflecting the microstructure of our cube. So here we just picked one fiber angle that was basically an average angle from all this data we collected. And then we build up this complexity in several cases until we reach case 7, where we had 3D Van Mises distributions for each of the slices that we took. And we plugged this in this hyperelastic constitutive model that was popularized and tested and validated by many people, including Michael Sachs and Holtzapfel, who've been fundamental in its development. So we can now fit this constitutive model, including all this microstructural data, to our experimental data. And so these are just a couple of examples of these 27 curves that we end up getting. And with this most complex model, we get a really good fit. And we're actually writing up this right now. And then you can look at all these tables of all the various cases that we tested. But you see that we, in the end, get a really good reflection of our material behavior. This is an example for the tension compression modes. And again, you get a pretty good fit here. But ultimately, we're interested in the entire ventricle, not just one tissue cube. So here we go back to our sonomicrometry data. So these are these fiduciary markers that we collected in the beating hearts of these sheep. And you see that these individual points are essentially these fiduciary markers. So now we can use spline surfaces to interpolate between these markers, and we get a representation for the epicardium, the outer surface of the right ventricle. And here you also see the reason why we actually replaced these sonomicrometry crystals with MRI dots before we did the anatomic scan. Because now we can actually take our anatomic scan and correlate these MRI dots from the MRI scan to our in vivo data. And as one of the examples, what we can do with this is we can actually get thickness maps from the MRI that we couldn't collect in vivo. And now we can create an endocardial surface from the epicardial surface based on these thickness measurements. We also have all these muscle orientations, right? So now we can use this MRI dot marker again to correlate these muscle orientations to a 3D volume. And because we collected data in the beating hearts, we also have the kinematic boundary conditions of the surface based on our sheep studies. And with this, we can build a 3D model of the right ventricle of a sheep. And so just as a reminder, this finite element model now is subject specific in that it has the micro and macro structure of that subject. The material parameters are informed from that subject. We have the kinematic and kinetic boundary conditions because we also measure pressure. And of course, the geometry is subject specific. One of the things we wanted to do when we collected these data was we wanted to have a good means to validate our model, at least the passive properties for now. And so what we actually did after we sacrificed these animals was we actually inflated the sort of arrested heart using a syringe pump. And what you see here is the right ventricular pressure that we produced with the syringe pump. So we inflated and deflated it. And that allows us to basically compare now our predictions to an actual measurement of the passive properties within that sheep's cavity, essentially. And so that's important because at that point, the tissue was as fresh as it could be. So the animal was alive a couple minutes before. And we can make sure that the properties that we derive from our material tests at least are within the right ballpark and haven't substantially changed through rigor mortis, say, or through the transportation process. So here you see our finite element simulation of this inflation process. And of course, we want to compare that now to the surfaces that we created from the sonomicrometry crystals. So here we overlaid the experimental surface with our predicted surface. And you see that there are some discrepancies, and we can quantify that in terms of error. But overall, they're very small and give us at least some sense of validation. Okay, so now we've developed this and, to some extent validated this pretty complex model. So we have just finished these essentially. And our next step is to actually undergo more rigorous validation and find a sort of, objective measure of how good is our prediction for these passive properties. We've done one of these sheep. We've actually collected data sets on 18 control animals. So we have lots of work to do this 18 times more. And we also want to identify the active properties. So far, we've only done the passive properties. And to this end, we actually will basically simulate an active contraction cycle of our ventricle and then compare that to our in vivo sonomicrometry data that we collected in these beating hearts to also identify active material properties. And then we repeated all this actually in sheep with heart failure, as well as sheep with heart failure and LVADs. So our goal now is to use our detailed model to really understand A, what happens in the right ventricle during heart failure, and B, what happens when we implant these left ventricular assist devices to really get to the base of what causes left ventricular assist device induced right ventricular heart failure. So that's the outlook for this project. Very intimately related to the right heart is the tricuspid valve as the key atrioventricular heart valve relevant to the right side. Similarly to right ventricle, I would like to give you some anatomic background, just so you know sort of what these terms mean that I'm gonna in a moment throw at you. You have to think about the tricuspid valve as essentially a check valve. Its function is to prevent backflow from the right ventricle into the right atrium, right? So you imagine the right ventricle contracts and the fluid will just want to go back through this orifice instead of going into the lung. So the function of the tricuspid valve is to close up this opening and redirect the flow toward the lung. It does this through the use of 3 leaflets, and these 3 leaflets essentially coapt and form a seal during systole, which means during the contraction. To prevent the valve from prolapsing, to sort of just flail through, there are these tendinous cords that essentially hold it back, and they attach to the right ventricle through these papillae muscles that you see here. So that's the essential function of the tricuspid valve. This is a histological stain or a cross-section of one of these leaflets, and similarly to the right ventricle, you have to acknowledge that this is a highly complex material. Regardless of what all these colors mean, what you see is in black is one structural element or constituent called elastin. You see it sort of layered in one area of the leaflet, but not somewhere else. In yellow, you actually see something called collagen, which is another structural protein. While in the middle, you kind of see this non-isotropic sort of amorphous mass called spongiosa. So it's a complex material, and it turns out just like the myocardium, and it's anisotropic, it's heterogeneous, nonlinear, viscoelastic. It can grow, it can remodel. And what's a real pain in the butt is there's 3 of them. So you have to do everything you do 3 times. So why go through the pains of doing all this? Well, just like with the right ventricle, little is known about the function, structure, and disease of the tricuspid valve. And that's pretty important because 1.6 million Americans have severe valve leakage. It turns out there's these crazy numbers out there of sort of population studies that find that actually 86% of us have some degree of leakage. And I recently got an echo, and it turns out I also have some leakage, and I'd like to think that's not a big deal. But in 1.6 million Americans, it's severe enough to affect their health. And so now it turns out that when we do repair it, which doesn't happen very often, so there's only about 8,000 surgeries for these 1.6 million Americans, that in 10 to 30% of those patients, the repair actually fails within a couple of years. So failing meaning that the valve starts leaking again. And we don't really understand why that is. And so again, I'd like to believe that a high fidelity computer model could help us optimize repair and improve the current outcomes. Just like with the RV, I'm going through this pipeline that we've developed to build this image-based and experiment-based model of the tricuspid valve. As opposed to the right ventricle that we studied in sheep, here we actually have a different model. We were very fortuitous to be collaborating with a team of transplant surgeons that actually got permission to use rejected transplant hearts. So these are hearts that were explanted from human donors, were transported to the recipient site, but for some reason were not being implanted because either the heart had defects that hadn't been known about, or the recipient was not doing as well as he would need to receive a heart. And so these hearts are in what's called an organ preservation system. So this organ preservation system is able to keep this heart alive in a beating state while recirculating the donor's blood. And we are allowed to basically do our pipeline on these hearts. This is a video from what this looks like. So this is a healthy human heart that's beating on its own. And we were able to implant these sonomicrometry crystals and then actually get the tissues from these hearts. So through this pipeline, we were able to collect sonomicrometry data on the right ventricle as well as the tricuspid valve annulus, and I'll talk about this in just a second. But our entire pipeline starts with us explanting essentially the leaflets after these hearts have been arrested. Based on these explanted leaflets, we can take detailed geometric measurements of the leaflet size. And remember, these leaflets are essentially these flaps that seal the orifice and basically fulfill the function of the tricuspid valve. And then based on these measurements, we can construct this three-dimensional representation of these leaflets. We, of course, can also measure the thickness. So we have patient-specific measurements of leaflet thickness. And next, we transport this to our lab and do the sort of standard test for these type of tissues, which is a planar biaxial test. Again, I mentioned it's kind of a pain that there's 3 of these leaflets. So we do this on 3 samples from 1 leaflet each. And we conduct this protocol that's a combination of off-biaxial and equibiaxial tests. So we get 5 curves per leaflet, and then we fit the sort of func type constitutive model to these curves. Here you see an example for these 5 different types of experiments and the simultaneous fit of these functions. And we see this classic J-shaped nonlinear stress strain behavior of these curves. It turns out that the chordae tendineae, so these are the strings that basically insert into the leaflets and prevent them from prolapsing, are critically important to the function of the valve and therefore should be modeled accurately. So now we also tested those. We tested at least 3 of those per leaflet, so a total of 9, but realistically we tested between 20 and 30 of them. And so we did uniaxial tensile tests, and here I sort of have one example from the posterior leaflets. These are 3 chordae, and we fit 1 curve, this is an Ogden model, simultaneously to these data. So now we have patient-specific leaflet properties that are also leaflet-specific, as well as chordae for each of the leaflets. In fact, for each of the regions of the leaflets as well. And I mentioned earlier that we put sonomicrometry crystals into the annuli of these donor hearts. That's important because it turns out that the annulus, and I remind you, the annulus is the interface between the leaflets and the surrounding myocardium, actually dynamically changes throughout the cardiac cycle. It contracts, it expands, and is very essential to the function of the valve. So we can track these points in 3D space and then actually fit a spline curve to it. So this is a representative cubic spline curve fit through 3 of these sonomicrometry crystals. And now we can take our leaflet geometry that I showed you earlier and actually wrap that around this annulus. And so that's how we create our subject-specific model of the tricuspid valve. Now, based on literature reports as well as pictures of our own valves, we can insert these chordae tendineae that enter sort of into this ventricular surface of these valves. And additionally, because we have the sonomicrometry crystals on the annulus throughout the cardiac cycle, we can also impose kinematic boundary conditions to the annulus. So we can actually have the valve contract throughout the cardiac cycle. And then we discretize this geometry, the chordae as well as the leaflets. And we can now run finite element simulations of this tricuspid valve. So here you see the closing behavior. And then of course we can report any sort of engineering metrics such as stress or strain and project them in all kinds of directions. But what I'm really very interested in is a metric that's not sort of the classic engineering metric, which is that of coaptation area. So coaptation is the process of the leaflets basically pressing against each other and sealing the orifice. And it turns out that clinically, that's really what surgeons or interventional cardiologists look at. Can your valve actually seal? They don't care about stresses in the leaflets. They care about, do the leaflets close? Do they contact? And how big is that contact area? So now we have this very cool high fidelity model in which we can really extract in detail which leaflets contact how. And that's really my primary quantity of interest that I will be studying with the help of these high fidelity models. And specifically, my interest lies in these medical devices called annuloplasty devices. These devices seem silly simply, yeah, in the sense that they're just these rings. They're metal rings, and they're being implanted on valves when they're leaky. And the idea is essentially that somebody designed the shape of these rings and said, well, every valve should look like this ring. So when we implant this titanium ring, the valve will take that shape and that will fix it. In addition, it usually downsizes the valve and sort of makes coaptation better. Or in other words, it increases the coaptation area. Turns out that we have learned that in 10 to 30% of patients after a couple of years, there's leakage again, right? On top of that, there's dozens of these different rings. So these are 3 different rings made by the same company. These are 3 rings made by another company. You see they vary significantly if you look at this ring versus this ring. On top of that, you see that the metal cores look very different. So this was a study we recently did, and we were very surprised once we open up these devices, how different they look. So our next plan is to implant these devices into our computational model. And now what's cool is that instead of clinical studies in which obviously you can only put 1 ring per patient, we can now iterate through all these different ring designs and sizes on the same patient and directly compare their success in improving coaptation area. So as I said, moving forward, we want to test all these rings. We've digitized them already. So this is just a matter of implanting them now virtually. We have 10 more of these patient-specific data sets, so there's lots of work to be done to also digitize them. As I said, we will virtually implant these devices. And what I'm very excited about is that we're not limited to one type of device. Transcatheter devices are sort of the new holy grail of the tricuspid valve community. So we have the opportunity to test all kinds of devices now in this high fidelity model that's based on true patient data. All right, I think I actually have time to talk about my last project, which I'm surprised by. So this is a recent project that one of my graduate students and I started in response to this COVID-19 pandemic. As a reminder, COVID-19 turns out to affect somehow the blood of patients and causes something called thromboembolism. So I'm going to briefly talk about what thromboembolism is. Blood has the ability to coagulate, to form a clot, and that's very fundamental to our well-being, right? When you cut yourself, you don't want to continue bleeding, you want your blood to clot. But sometimes that happens in the midst of our vessels, right? So this is sort of a cartoonish version of what that looks like. And now when that breaks off, it will travel downstream and occlude some artery or some vessel. If it occludes your pulmonary artery, then you have what's called a pulmonary embolism and you can't breathe. If it occludes a cerebral artery, then you have an ischemic stroke. And if it occludes a coronary artery, you have a heart attack. And fascinatingly, this is essentially a fracture problem. So one of my questions was, would it be able to actually test the fracture toughness of clot to predict whether it's likely to break off or not? And this may seem like a crazy idea, being essentially we take the blood from a patient, we clot it, we pull it apart, test its fracture toughness, and make some sort of prediction of the risk that this patient experiences. But it's not that crazy in that mechanical testing of blood has a long history. And so essentially bleeding disorders were first diagnosed by measuring how long it takes to coagulate blood from that patient. And that was done by mechanical means. In other words, you know, you can either test the viscosity of the blood mechanically, or actually the first sort of industrialized version of this test was 2 rakes that were pulled apart. And only when they could sense a resistance, it was determined that the blood had coagulated. So mechanical testing has a long history for being used in blood diagnosis. And so sort of this crazy vision of mine is to now miniaturize this kind of test and then be able to sort of develop a patient-specific risk factor for thromboembolism. So I have no training in fracture mechanics, but in my department, that's essentially what everybody does. So the first thing they told me is like, oh, you know, you do a mode 1 pure shear test, which we did. And then I realized that this is an incredibly complicated material, and we don't have the data to actually inform inverse computational models to back out fracture toughness. So the first thing we did was we developed a test to essentially characterize the sort of non-surface creating dissipative mechanisms that may be contributing to the fracture toughness, effective fracture toughness of materials. And so what we did was we kind of did these classic tests that surprisingly haven't really been done in this simple manner, which is we kind of tested the strain rate dependence and found that blood clot is strain rate dependent, although not that significantly. We also found that blood clot experiences Mullen's effect as well as hysteresis and permanent set. For example, after 1 loading cycle, we have 12% of permanent set. blood clot actually undergoes the sort of preconditioning response or the sort of equilibrium. So if you cycle it 10 times, it turns out that it changes. And when we compare the first cycle to the last cycle, we find that blood clot after 10 cycles has become an essentially different material. So we've learned this about this material. And we also learned that blood clot actually shows nonlinear viscoelasticity in that the relaxation time actually increases with strain under this sort of stress relaxation test. So our goal now is that we've developed this basic understanding for these dissipative mechanisms, a computationally based inverse algorithm to now perform these mode 1 experiments using, say, cohesive elements and then back out fracture toughness. And we are just in the process of getting permission to use patients' blood to first prove on healthy patients that we can do this, and then perform this also on blood from patients that have had or currently have COVID-19 to see whether we actually see a difference in fracture toughness that could explain the higher risk of getting thromboembolic disease. I skipped a couple of points here. Essentially, we also want to do some basic science to understand what are the blood constituents that actually contribute to fracture toughness. But ultimately, our goal is to see whether we can use fracture toughness as a predictor for blood disorders. With that being said, many thanks to you for listening and being here, some of you a second time. A million thanks to my graduate students, Ella, Will, Sotirios, and Rodang, and a bunch of undergrads who've really helped out. Thank you to collaborators like Adrian, who's been hosting me today, Jeff, whom I've seen on this call, as well as my clinical collaborators, and also the funding sources that have helped a lot, obviously, with these projects, as well as my department, who's actually funded this thrombus research. With that, I'm open to questions, and again, appreciate you being here.
Thank you very much, Manuel, for the very interesting talk. I really enjoyed it, as I'm sure all of the members of the audience. So please, just a reminder to post your questions to the chat, and then I will read out the questions, and then one of the staff members will unmute the person that is asking the questions in case they have some follow-up questions. So the first question that we have is from Alexander Kaiser, who asks, what is the role of heterogeneity in tricuspid mechanics? There's quite a bit of variation in local thickness, especially around chordate insertion, thicker bundles by the free edges, and so forth. How much does this matter, and how well understood is it?
That's a rich question. I think in general, we can say that spatial heterogeneity is very important. I think in general, we can say it's hard to capture it. And in general, we can say we should have done it, but didn't do it. So yes, it's really true. We're ignoring this right now, given the intense sort of complexity of the project that we've already looked at. But in fact, we have an optical coherence tomography device in our lab, and I bought that specifically to capture at least the heterogeneity in thickness, which I assume will make a huge difference. And I think the question is really insightful in that, yes, the chordal insertion sites are very different from the rest. And I think characterizing the valve leaflets as these sort of smooth, homogeneous membranes is just outright wrong. And I think it is, our burden to kind of estimate how big the error is that we accrue by ignoring it. And hopefully, as we get better at all this, we're going to start including that and getting better models.
Thank you. Yeah, I don't know if Alexander Kaiser has any follow-up, or is it good?
No, that's very consistent with my opinion, and also reading your old papers about the mitral valve. So so it's interesting to hear your further thoughts on it, and also that you think it's a valuable avenue to investigate in the future. So thanks.
I really appreciate it, and thanks for reading my old papers.
Yeah.
We have a question from John Walmsley, who asks, one of the major anatomical features of the human right ventricle is that it is heavily trabeculated on the endocardial surface, and trabeculae are a significant proportion of RV mass. However, almost all computational approaches to the RV disregard these features and treat the endocardium as smooth. To what extent do you think we should be worrying about these features?
I think my answer is entirely in line with my answer to the previous question. You're absolutely right. It's probably important, and yes, we ignored it. So our MRI scan would have captured the trabeculation to the extent that our spatial resolution allowed for it. However, we entirely ignored that in our simplistic reconstruction of the geometry. There have been actually, there's both surgical approaches that have actually ablated the trabeculation to look at what that does. And there have also been studies to use that as a therapeutic approach toward reducing the effects of hypertrophy. I think we are in the position to actually study that, and that we also have the true RV geometry. And I'm very curious to see what the effect is. But just as my answer to the previous question was, I hope we get there as we sort of overcome even more simplistic challenges that we're kind of still facing. But absolutely right, and we should include it, and hopefully in the future we will.
Are there any follow-up questions from Andrew Seifert? Sorry, from John Walmsley.
No, I think that covers it really. It's just something that's quite striking when you look at these models, but it's never there.
It's absolutely true. I appreciate that. I really do.
The next question is from Andrew Seifert, and I apologize if I mispronounce the names. And the question is, the ability to simulate leaflet overlap within the tricuspid valve model is novel and exhibits significant potential, namely because in the clinical setting, actually measuring leaflet overlap is quite difficult. Because of this, the questions are, how many samples do you believe you will need to create a population averaged model? And two, is there potential for this model to simulate leaflet approximations?
I appreciate that question. I think my answer to the first part is, I don't think we'd necessarily want to get a population average model. And I think that's something that I will want to get at with the data that I have, that all these approaches, these devices that are built for population average models, and that means that on average, they probably do well. But that also means that we have a significant number on both sides of the distribution that probably don't do well with it. And what I would like to use my data instead for is to demonstrate that averaged models may not be most optimal for everyone, right? So that's sort of my first answer. The second one is, yeah, absolutely. We have the potential to look at leaflet approximations. And as I said in the sort of last bullet point of my outlook, I'm extremely excited to have this platform with which we can test all these ideas, including approximations and novel devices, etc. And I think I would like to highlight that the FDA is actually moving toward acknowledging the potential of computational models in their process of evaluating risk and efficacy of medical devices to save animal lives, as well as protect the patient population from sort of premature clinical trials.
Is there any follow-up from Andrew Seifert? Nope. All right. I do have a follow-up actually to that question, which is, you've been doing these measurements, for example, from the tricuspid valves for different patients. How much variability is in there? So you're saying that average models may not capture correct, they may not work for every patient. And is that because of the variability that you see? And how much is that variability essentially? Because I think that would dictate how bad or good these average models would be.
I love that you were able to pose this question without asking for uncertainty. I think the uncertainty is huge in these data. And actually the valve that we started out with, am I able to go back? No, I just screwed that up. It doesn't really matter. The valve that we created, if you are a person who looks at tricuspid valves, doesn't look at all like your sort of, textbook tricuspid valve. In fact, the septal leaflet is too small, the posterior leaflet is distorted. And I fully anticipate that when we publish this, we will get reviewers that say, this doesn't look like a tricuspid valve, because we've all learned that a tricuspid valve looks one way, but it really truly doesn't. The reality is that we've been exposed to so many population average data that we don't actually really appreciate the diversity that we see in tricuspid valves. If you look at sort of, the next person over, there's so much diversity in sort of human anatomy and human expression, right? The same is true for a heart valve. To a point where we today don't even agree anymore that it's a tricuspid valve, and that some people have 4 leaflets, some people have 5 leaflets. So I think the uncertainty is huge. And yes, I 100% believe that it's that uncertainty that leads to population average approaches being unsuccessful.
I have a quick follow-up questions and take people's time. But the other question that I have is, how important is it? Are you thinking about doing a sensitivity analysis? Because maybe these variations are actually not important. So how do you determine how important they are?
How do I determine? Yes, I mean, 100% that's our goal. So as you know me quite well, I love experiments and I love sort of, solid experimental data, but I'm really moving toward having to appreciate sort of model reduction and uncertainty analyses and sensitivity analyses. And that's really our goal moving forward. These simulations are very expensive right now. And at least the sensitivity, sort of global sensitivity analyses that I've looked at are very expensive. So we're looking at a way right now to reduce the cost of these simulations so we can do global sensitivity analysis and do understand how important is it that we get the geometry of these leaflets right? How important is it that we include tissue heterogeneity? And we also go to answer a lot of these questions that were asked in the course of this Q&A, right? We actually don't know how important it is that I ignore thickness distributions of the leaflet, but we will find out, and sort of we're starting to do exactly that. And I think our data set will be a great platform and framework to do that.
All right, thank you. I have a question from Rob McLeod, and again, I apologize if I mispronounce the names, but the question is, there are other clotting disorders besides the COVID situation? Have you thought about this range of diseases as another worthwhile space? And
100%. I'm an opportunist, like the next person, in the sense that I'm trying to sort of, apply with this approach to sort of these rapid requests for proposals, et cetera. But ultimately, my interest actually was sparked earlier in that thromboembolic disease is actually the #1 cause of death. 1 in 4 deaths are due to thromboembolic conditions. So that includes ischemic stroke, that includes myocardial infarcts, that includes thromboembolisms. So absolutely, there's a huge range. And this idea, the few people that kind of know about thromboembolism, they think, oh, this is sort of the disease of sort of the old, it's a disease of people with, pre-existing conditions. And that's actually not true. In the industrialized world, it's actually the #1 cause of death in pregnancy. So this is a huge issue, and we're 100% motivated and interested in kind of looking at a broader spectrum of patient populations.
Is there any follow-up from Rob McLeod? I think we are in the process of unmuting in case there's a follow-up question.
Yeah, no, I just wanted to overall, thank you, Manuel. Great to see you again. Fantastic talk. I love where this research is going. We have many things we should be talking about. And I can assure you that there are clotting diseases that are inherited, that I have one that first showed up in my 30s. So, and it's something I take medications for every day because it's caused several blood clots and even pulmonary embolism. So there's a whole family, and they're poorly understood, poorly characterized, and there's no, there's some okay tests that tell you something about them, but they're not very quantitative and not very helpful. So it's a really important area.
Rob, have you thought about donating blood to the University of Texas at Austin?
I'd be happy to give blood to anyone who wants to examine it. They don't want it, it's not normal enough anymore to be used for transfusions for injury people, but yes, I'd be happy to provide blood for.
But joking aside, I think what's challenging about it is that the general strategy right now is to classify people as at-risk populations, and they're all treated equally, right? And the drugs you're given are not good for you, right? Your blood is supposed to coagulate in certain situations. So if you prevent it from doing so, you suffer from bleeding disorders. Actually, your risk of ischemic strokes goes significantly up. So if you had a measure that can determine that one person in a subject-specific way is more likely to have an embolism, that would be a huge advantage in that we could spare the other people from these sort of general, pretty drastic side effects that you experience with this antithrombotic regimen of drugs.
Yeah, no, absolutely right. Right now it's trial and error, and hoping you don't have a bike crash, while you're on the drug. Thanks.
Appreciate it. Thanks, Rob.
Okay, we have a question from Emily Johnson, who asks, could you further explain how the chordae attachment locations and groupings are determined for the tricuspid valve model? Is the motion of the papillary muscles considered in the TV model? If so, how is this motion captured?
I feel like Emily must have listened to a lab meeting in which she discussed all the weaknesses of our model. These are really good questions. And it's probably honestly the biggest weakness of our model. It's very hard to model a chordae because it's such a mess. If you do mitral valve work, you're actually sort of lucky in that there's quite a bit of organization in the chordae, so they all kind of insert into papillary muscles. In the tricuspid valve, it's a mess. So they insert into several papillary muscle heads, they insert into the endocardium, et cetera, et cetera. So in the end, really what we did went through a pretty painful trial and error in which we inserted chordae sort of where they have been described to insert and where we sort of seen them insert. And then we literally ran simulation after simulation until we basically enabled closure of the valve where the free edges would line up, right? Because we know the exact geometry of the leaflets. And we essentially argued there wouldn't be sort of an overlap, right? Not one leaflet would sort of co-op with another leaflet like this, because if there was no load on that free edge, it would probably degrade over time in this sort of, general sense, oh, I'm not being used. So that's what we used essentially as a metric to sort of lengthen and shorten chordae, insert them somewhere else. So that's definitely the weakness of our model. And that's something that we have to acknowledge as a limitation. We do have 2D echo from our experiments, and we use that in a somewhat qualitative way to make it sure that sort of the general cooptation lengths, angles, et cetera, matched up. But it's not as rigorous as I would like it, for sure not.
Is there any follow-up from Emily? Emily Johnson.
Thanks for the explanation there.
Oh,
yeah,
for sure. If you have more questions, please feel free to shoot us an email. We love talking about the tricuspid valve.
Thanks.
Moving on to the next question by Gerard Teschen. Says, nice talk, Manuel. When you present the results on the cubic sample of the RV wall, you mentioned exploring different levels of complexity. What do you conclude? Do you need the least or most complex model?
I love that question, and I really wish I could answer it very satisfyingly. So we had 4 samples, so that's not a lot of data. But we actually went through this, and I 100% anticipated that the most complex model will give us the best fits. It turned out it really didn't. It turned out that among the 7 cases, it was actually the simplest case that gave us the best fits. And we were pretty discouraged by that and couldn't really explain it. And we went through a lot of sensitivity analysis because I felt like we had done something wrong. And I asked my student to go back over and over again to make sure that sort of on the modeling side and on the math side, everything was right. And I talked to a friend of mine, Emma Lejeune, and she suggested something that actually turned out to be really cool, which is we went back and actually fit all 7 cases to only 3 samples. And we found the same thing. It doesn't really matter how complex the model was, the fits were about the same. They were all pretty good. But then when we predicted the fourth data set, it turns out that the most complex model gave the best predictions. So essentially what that says is that by including the richest amount of data in our microstructural approach, we made the best predictions. And so I think that's a pretty valuable lesson to be learned about the difference between, fitting our data to all our data sets versus, using a training set and trying to predict an unseen data set and sort of highlight maybe the strength of detailed microstructural data. Does that make sense, Gerard?
Yes, thank you, Manuel. That's actually a very nice finding that the predictions are improved. I appreciate that outcome, and it's something that you should report, if you have maybe a larger data set to demonstrate it.
Gerard, maybe you say that one more time because my student is on the call and I've been waiting for the manuscript, so.
Manuel, you must get this work done.
Thank you, Gerard. I'll forward it to my student. Thank you, I appreciate it.
Sure.
We still have a couple of minutes for questions. So I'm gonna read the next one, which is from Reza Abbas, who says, great talk, Manuel. I have 2 questions. One, does RV remodeling contribute to the tricuspid valve leakage? And the other one is, does your cooptation area metric address the short-term success of the surgery, or it can be considered as a long-term metric as well?
Amazing questions. I don't really know how to answer them. I think that reflects on how good of a question it is. I think the first one I can say yes. Reza, the mitral valve, fails functionally and organically. The tricuspid valve something like 86% of severe tricuspid valve leakage cases are functional. So the tricuspid valve essentially fails functionally, and that means that it's other factors than valve inherent factors that make it fail. And that means it's usually RV dilation. So yes, RV remodeling essentially causes TV leakage. I find it a complex question to answer because I've invested probably 60 to 70% of my resources over the last 3 years trying to actually demonstrate that it's not just that this idea that the tricuspid valve fails functionally is not really true, and that the tricuspid valve remodels in functional disease. And that's important because that means that we haven't really understood it to begin with. And that also answers your second question in the sense that, no, it probably won't predict it because changes to the valve likely cause changes in mechanobiological equilibrium of the leaflet tissue, which likely makes it stiffer and fibrotic and changes it altogether. And I believe that that really contributes to valve leakage as much as other factors, which is entirely unexplored at this point. We just submitted this paper, so fingers crossed that that goes through and we can sort of claim this officially. But I think over time, coaptation area is probably not a good predictor, and there's probably other metrics that we should consider as a sort of predictive metric.
All right, thank you, Manuel. It was great.
Oh, for sure, thank you.
I have probably time for one more question, if there's any. Manuel, you gave us enough time for a very, very long discussion, which is great.
Yeah, for some reason, I was so anxious about not getting through it, and then, I did.
Well, I guess due to the second try. Okay, there's one more question from Alison Poach. It says, great talk, Manuel. I find tricuspid valve studies particularly interesting. How do you think your experiments can help surgeons optimize repair in terms of ensuring there is enough leaflet coaptation to prevent regurgitation while also making sure the valve isn't over-restricted?
I really appreciate it, Alison. I think you answered your own question in a way, right? I think what we can do is we can help them ensure that there's enough leaflet coaptation and the leaflets aren't overly restricted through computational simulations. But in seriousness, I think what the value of our simulations ultimately will be is that we have real patients, right? And we have, representations for valves of real patients that are informed by real patient data. So we have a lot of fidelity or confidence in the quality of these models. And we also have a decent representation, not a huge one, right? We have 10. But we can now really go through this process of trying these sort of like standardized approaches on these valves of a pretty representative population. So we have one patient who was 54 kilograms. That's like, 120 pounds. And we had a 250-pound patient. We had a pretty good representation between women and men, a good representation of age. So I hope that we can really, moving forward, basically run a device through all different scenarios and derive metrics like coaptation area or leaflet restriction to optimize approaches. But I think the vision is to move forward and kind of be able to do that. Obviously, this sort of on the run, right? 3D echo has given us the capacity to really visualize the valve in 3D. There's your work and the work of many others that tries to kind of take a direct pipeline from this 3D representation to final element models or other numerical methods to kind of make predictions on the run, sort of live to the surgeon or intervention cardiologist as they try to fix the tricuspid valve. And I think that's the way to go. And I think that's a vision that we all share, and that's where I'm sort of headed with this approach. And hopefully our data will become sort of a atlas that we can use to improve our predictions based on only a pretty high noise sort of 3D representation that we took in the surgical suite.
Thanks so much, Manuel. This is really exciting work. As you know, I'm not a surgeon, but I think so often surgeons really try to optimize that coaptation area to the point where sometimes when I look at the post-op models, it looks like the valve really is no longer in a physiological state. It makes me wonder if the overcorrection is actually really what has some negative impact on repair durability. So I'm really excited to see this presentation and looking forward to seeing where it goes.
You're 100% right. And I appreciate so much that you say that, in that I think what we can add is more than just leaflet area, right? We can also add stresses. And I'd really like to explore, and Michael Sachs has done this, for example, on the mitral side and many others. What is the impact of changing the mechanobiological equilibrium of this tissue to long-term repair stability, right? It's not just sort of dehiscence due to large stresses. It's also, how does the tissue react to just being deformed completely differently than it was before? And I think you're right in that maybe trying to optimize just this one metric while sacrificing, stresses or any sort of other engineering metric may contribute to this long-term failure that we observe in these patients, particularly on the tricuspid side.
Right. I would like to thank once again Manuel for giving us this interesting talk, and also all the audience for engaging in this discussion following the seminar. So I think I will just follow Gerard's attention and virtually clap. And thanks everyone for being here on behalf of USACM, and I had to step in today in lieu of John Doble, but I'm sure you would have been excited to see this presentation. And I think this concludes the seminar today, and actually concludes the summer seminar series. So I think this has been quite a success. So I'm glad to be the one that sees it to its end. So yeah, great talk, and yeah, nice seeing everyone, and stay safe.
Appreciate it. Thank you so much again for the invitation. Thank you, Adrian. Thank you everyone for being here.